Uh, Thank you, Sebastian. Okay, hello everybody. Today, Mojca and I will present some basics from the topic of the wastewater treatment using advanced oxidation processes. We both come from the University of Ljubljana, from the Faculty of Mechanical Engineering, and today, in the scope of the lectures, uh, we will take a look at wastewater, what wastewater actually is, and what kind of pollutants can usually be found in them, how wastewater treatment plants actually work. We will put a little bit more focus on the pharmaceuticals and what are the techniques, how to deal with them. We will take a look what the legislation say about their presence in the environment. Um, since our department is generally focused on cavitation studies, we perform many research studies on utilizing cavitation for wastewater treatment, for which some case studies will also be presented today. So, what wastewater actually is? Commonly, the term wastewater is used as a synonym for sewage, but in reality, it is divided into domestic wastewater, industrial wastewater, and so-called storm runoff wastewater. We can imagine that domestic wastewater contains household gray and black water, while industrial wastewater depends on the specifics of individual processes used in the factories. And storm runoff wastewater is water that comes down on the streets due to rain and snow. So general types of water pollutants usually includes pathogenic organisms, oxygen-demanding wastes, plant nutrients, synthetic or organic chemicals, inorganic chemicals, microplastics, sediments. It can contain also radioactive substances, oil, and heat. If you take a look, oxygen-demanding waste is an extremely important pollutant to ecosystems. Most surface water in contact with the atmosphere has a small amount of dissolved oxygen, which is needed by aquatic organisms for cellular respiration. Decomposers such as bacteria and fungi also conduct cellular respiration and consume oxygen as they break down the dead organic matter. Too much decaying organic matter in water is a pollutant because it removes oxygen from water, which can kill fish and other aquatic animals. And heat, for example, is also considered as a pollutant because rivers are often used in many industrial processes for cooling, for example, in power plants. And as one can imagine, an excess heat can do a lot of harm for aquatic environment. Thus, there are regulations and restrictions for how much water that is used for cooling can be heated. So you cannot heat the river, for example, above certain degree for certain amount of certain degree of Celsius. In the past, wastewaters were directly discharged into the environment. Actually, in Roman times already, around 600 BC, a first known modern sewage system was built in ancient Rome, and it was called Cloaca Maxima. And that sewage system improved the general health of the people greatly. And removed large amount of waste and wastewater from the city. The information that I found on the internet, but I cannot be sure whether this is 100% sure or not, since the number is very high, but nevertheless, according to the web page, nature.org, it is estimated that 80% of the world wastewater enters our environment untreated. So this number is pretty high. And we in Europe, we live in modern cities, and Modern cities usually used wastewater treatment processes to treat wastewater before releasing it into the aquatic environment. And since the population is growing, the amount of wastewater that needs to be treated is also growing. Thus, it's extremely important that more focus is put on the research, how to deal with all the pollutants that are released due to all our human activities. So here on the slide, we have a schematic presentation how a conventional wastewater treatment plant actually works. Here we can see a typical wastewater plant and its stages. Conventional wastewater treatment usually consists of 3 stages: preliminary stage, primary stage, and secondary stage of treatment. After preliminary stage, where bigger materials such as tree limbs and leaves are mechanically removed from wastewater influent, the treated sewage enters primary treatment zone, in which physical treatment referred to as sedimentation is generally utilized to remove sandy material and other particulates and oils. Secondary treatment step is an aerobic treatment step. Here, dissolved organic material is oxidized and removed through biological processes. The wastewater is mixed and aerated in special tanks or series of tanks if the wastewater treatment plant is big. And in these tanks, population of different microorganisms, mostly bacteria, uses different dissolved electron donors, for example, oxygen, nitrate, and nitrate ions to oxidize substrate, which generally leads to increase in the bacterial population through reproduction and transformation of organic compounds. Advanced sewage treatment process usually include also tertiary treatment step. Its purpose is to disinfect the water before releasing it into aquatic environment in river or oceans to kill bacteria and microbial pathogens. Most commonly used techniques are chlorination, UV treatment, or ozone treatment. Sludge that comes from clarifiers is settled. This sludge is called waste-activated sludge, and it usually goes through anaerobic digestion in the absence of oxygen, where anaerobic bacteria digest it and produce biogas. With this process, the volume of the sludge is reduced and some energy is restored. Digested sludge is most commonly due to presence of heavy metals and other pollutants incinerated instead of left on landfill. For quaternary treatment, which involves removal of other micropollutants such as antibiotics and hormones, this quaternary treatment is currently under consideration to put in legislation for wastewater treatment because such micropollutants are only one part of contaminants of emerging concerns, for which we will present in the next slides. So please, Mojca, go ahead.
Yes. Hello. So now that you have heard about the water treatment, let's go into the contaminants of emerging concern. These are by definition chemicals that are present in water bodies and are not yet regulated, but pose danger to the environment, including humans. These compounds are not necessarily new chemicals, but have been addressed as critical environmental pollutants only in the last decades. And the growing concern is because their fate, behavior, and adverse effects in different ecosystems are not yet fully understood. And they include compounds such as pharmaceuticals, personal care products, industrial chemicals, PFAS, and pesticides. And the biggest problem that we have is that in parallel with the growth of the world's population and its standard of living, also production of these substances is increasing, which leads to increased pollution. And hundreds of thousands of tons of these compounds are produced, consumed, and dispensed annually. And since this growth will not abate in the near future, the burden on the environment will only grow in the future. Pharmaceuticals, for example, pharmaceuticals, for example, have been present in the environmental, in the aquatic environment for a long time, but they have found their place in the scientific and popular press as important environmental contaminants only from the mid-90s onwards. And their recognition as environmental pollutants was mostly due to the advances in analytical instrumentation and isolation procedures, which allow detections of relatively low concentration of these compounds within the nanogram per liter to microgram per liter concentration range. In target organisms, most pharmaceuticals undergo metabolic transformation, which decreases their pharmacological activity and facilitates their excretion. And due to the metabolism, the parent pharmaceuticals can be excreted as unchanged compounds or as a major metabolite or as a group of many different metabolites. So what we are faced once they are excreted is that we have a mix of known and unknown compounds, and their modus operandi in such cocktails is far from being understood. And the fact is that pharmaceuticals are present in the aquatic environment into which they can enter through various pathways. And up until now, more than 160 pharmaceuticals belonging to different therapeutic classes have been detected in the aqueous environment. So what are these environmental exposure pathways through which they can enter the environment? So we can have a direct or indirect input. And examples of direct input are after veterinary use, where they are used for therapy in aquaculture or agriculture. And from here, they can enter the surface or groundwater directly. And two examples of indirect input is after they have been used for therapy, or when they're improperly left disposed of in the landfills. So in this indirect use for therapy use, a mixture of these unaltered pharmaceuticals and their metabolites can enter from the environment, from hospital waste effluents, household drains, and from pharmaceutical production facility. They reach wastewater treatment plants through the sewage systems. And since the conventional water treatment methods often prove inadequate to address the intricate nature of these pollutants effectively, they enter the aquatic environment, including groundwater and drinking waters, via these wastewater treatment effluents. And pharmaceuticals were detected in all these different mentioned receiving waters, thus surface water, groundwater, drinking water, in the concentrations ranging from nanogram to microgram per liter. And despite a lot of data that has been gathered so far and piles of proof of their presence in the environment, for most pharmaceuticals, the decision whether they pose a significant threat to the environment is still due. However, in response to the growing concerns about water pollution and to ensure a sufficient quantity of good quality water that is available across the entire European Union, a watch list of priority substances to be monitored in the EU surface waters has been established within the Water Framework Directive in 2001. And through the years, the watch list has constantly been amended and new compounds are being added to the list, including more and more pharmaceuticals. The first pharmaceuticals to be included in the first watch list were diclofenac and two hormone disruptors, estradiol and ethinyl estradiol. And afterwards, other compounds followed, such as antibiotics and antimycotics. And all these monitoring of substances in surface water and determination of their toxic effects has led the legislation to start questioning what to do in the future in order to prevent or to eliminate these compounds from the environment. And thus the quaternary treatment, the legislation regarding this removal will be put into motion presumably in 2025. Where quaternary treatment will be will have to be put into some wastewater treatment plants. Once pharmaceuticals reach wastewater treatment plant, the most important treatment step for their removal is in the secondary treatment step, where the biological treatment is involved, and the oxidation, as was mentioned before by Martin. The fate of pharmaceuticals in wastewater treatment strongly depends on their physical-chemical properties, thus log-Kv, dissociation constant, biological degradation rate constant, and sorption constant. And the result of what happens with them once inside the biological treatment can be mineralization, which means that the substance is degraded into two inorganic products, CO2 and water, which is the most preferable degradation pathway. They can be adsorbed to solid particles, biodegraded or biotransformed into inactive or even more toxic products, or they can enter wastewater treatment plant completely unaffected by all processes. Unfortunately, a lot of pharmaceuticals are recalcitrant, which means that they go untreated via the wastewater treatment plant. And for this reason, wastewater treatment plants are considered as a major source through which these pollutants enter the environment. And to tackle this problem, the wastewater treatment plants need to be upgraded or retrofitted with some alternative processes. The investigation of these processes is now even more urgent than ever if the quaternary treatment will play a role in the future. So mostly investigated advanced or alternative wastewater treatment techniques include photochemical and chemical processes and mechanical processes. Again, I have to point out that the same as was true for the biological processes, also for these advanced treatment processes, the susceptibility of these compounds to degradation with these treatments strongly depends on their physical-chemical properties. Photochemical and chemical processes are based on elimination of organic compounds by structural transformations, which can be achieved by several different mechanisms. And mostly investigated chemical processes include photolysis, ozonation, and advanced oxidation processes. Direct photolysis involves interaction of artificial light with a target molecule, and absorption of photons by target compounds causes induction of photochemical reactions, which can lead to degradation of target compounds into intermediate products whose further decomposition can lead to total mineralization. Another chemical process which is important is ozonation. And ozonation, as mentioned before by Martin, can be a part of tertiary treatment used for disinfection, but can also be an advanced treatment. As an oxidant, ozone can oxidize organic compounds directly or through formation of hydroxyl radicals that are produced when ozone reacts with water. And the third option, and in the last decades mostly investigated, are advanced oxidation processes, which are processes where powerful oxidizing species, namely hydroxyl radical, are formed in situ, and these radicals can non-selectively attack organic compounds, and preferably, again, resulting in their complete mineralization. And over the years, sufficient data have been collected to confirm that advanced oxidation processes indiscriminately attack many structurally diverse hazardous organic pollutants, leading to their partial or complete oxidation. And the processes that are investigated and where the OHA radicals can form in situ are, for example, phenton oxidation, electrochemical oxidation, UV combined with hydrogen peroxide, gaseous plasma, and photocatalysis. And of late, a lot of investigation is also dedicated to cavitation, hydrodynamic, and acoustic cavitation?
Cavitation is a physical phenomenon which can only appear within liquids. It is similar to boiling. If you imagine a cup of water that is put on a stove, what happens? The temperature of the water rises at constant pressure, atmospheric pressure, and it boils, and it starts to boil at approximately 100 degrees Celsius, depending on the atmospheric pressure. And everybody knows that. But if you take the same cup of water at room temperature and put it in a closed container and suck the air out of it, the temperature will stay the same, but the water will eventually start to boil due to decreased pressure. At what pressure it will boil depends on the temperature of the water. And the cavitation actually describes a formation of small gas vapor bubbles that are generated due to locally decreased pressure. It is most commonly observed on water turbine machines, such as ship propellers, water turbines, or water pumps. If you imagine, if the blade of the ship propeller goes through the water fast enough, the pressure behind the blade locally decreases, and if it decreases slow enough, the water actually boils there. But when the pressure recuperates back to the initial pressure, these bubbles collapse and cause various negative effects. On this video here on the up left corner, we can see cavitating flow in a simple Venturi constriction. We can see that cavitation structures, bubbles, are formed behind the narrowed part of the constriction and started to collapse in the divergent part. What happens when the bubbles collapse? You can see on these two videos below. Here we have cavitation bubble formed due to electrical discharge in the vicinity of the solid boundary from the side and front view. We can see that the bubble collapses asymmetrically towards the wall, creating a so-called microjet, and due to high velocity and shock waves causes damages on the surface, which can be seen on the video on the right side. And when the bubbles collapse at the center of the created bubble, we can get extreme pressures, extreme velocities, extreme temperatures. And all these are causing negative effects like noise, vibration, reduction of efficiency of the machines, and what we engineers are most scared of, the material wear or so-called cavitation erosion, which can be seen on the propeller on the bottom left side. In general, we can divide cavitation by source of the local pressure drop, and we distinguish between hydrodynamic and acoustic cavitation. Hydrodynamic cavitation is caused on rotating machines like ship propellers, water turbines, or in the flow tract, where we have certain constrictions where the flow is accelerated and the pressure drop is a consequence of this flow acceleration, like in a nozzle of a fuel injector. Here on the right, we can see a cavitation vortex generated at the outlet of the Francis water turbine. And this vortex, for example, is causing serious vibration on the whole turbine. Here on the video, in the middle, we have a cavitating flow in a shape of von Karman vortex that is generated behind the cylinder in the flow tract. Another type of cavitation is acoustic cavitation, where local pressure drop is generated due to acoustic waves propagated through the liquid. For example, one of the most commonly used acoustic devices is ultrasonic homogenizer, like this on the image on the left. And what happens under the tip of this ultrasonic horn, we can see on the bottom left video. We can see that cavitation, that the horn is oscillating up and down with relatively high frequency in terms of several kilohertz, and water beneath the tip of the horn evaporates. So we mentioned that cavitation has many negative impacts, especially on the surfaces where the cavitation bubble collapses. So the idea is whether cavitation can damage the material like steel. Can we also use it to destroy bacteria, viruses, or even something else? And the answer is yes. Due to extreme effects that we mentioned, we usually divide these effects between mechanical or physical and chemical effects. With these extreme effects, we can cause, for example, bacteria to rupture, virus to inactivate, or even, as Mojca already mentioned, we can achieve oxidation of different chemical compounds, because due to the extreme temperatures that can happen when the cavitation bubble collapses, the water molecules that are there can dissociate into OH and H radicals, and these radicals are extremely reactive and can oxidize various chemicals. We also made a research study where we determined how many radicals are formed within the cavitating flow. For this, we used a salicylic acid dosimetry, and along the way, we also discovered that cavitation was strongly influenced by extremely small amounts of added salicylic acid. And the result of this study was actually that one needs to be extremely careful when using various chemical methods for determination of, for example, chemical effects by cavitation, because we noticed that even really small amount of added substances in the cavitating flow can change the cavitating flow completely. Here on the image in the middle, we can see on the upper image we have pure water, and then we increase the amount of salicylic acid, and this is in milligrams per liter. And we noticed that cavitation was completely altered visually and also acoustically. So all of our investigations are made on cavitation devices that we designed and made by ourselves within our department. And for various purposes, we designed cavitation devices where we can treat sample volumes from 10 milliliters to 100 liter. We usually divide them into static devices, which are designed in a way that the liquid is pushed through the constriction, usually through simple Venturi constriction or an orifice plate. We also designed, in principle, two types of static cavitation devices. One type where we use a pump for sample circulation, and the other where we use two reservoirs and we push the sample through the constriction between the two reservoirs, either with compressed gas or with pistons. In some cases, we try to eliminate the possible effect, the potential effect of the pump that is used for transport of liquids. And that's why these designs where the liquid or the sample is pushed through the constriction with the compressed air or the piston help us to go more into depth in understanding what actually is going on with our cavitating flow. So on the videos on the left, we can see, for example, here we have two constrictions, two orifices with 3 and 1 holes, and we can see that by changing the geometry of the orifice, for example, we can change the cavitation conditions that are generated in our devices. Besides static cavitation devices, we also put a lot of effort into the development of so-called dynamic devices with the emphasis of a future scale-up. These are rotating generators of hydrodynamic cavitation. We have different designs on lab scale or even on a pilot scale. One of the big advantage prior to other cavitation generators is that our devices have double function. They work as a cavitation generator and as a pump in the same device. And for that, we hold a few patents for them already. And the idea is to develop and adapt each device for the specific needs of treated samples. So depending on the pollutants that we want to treat, we adapt the cavitation so that it has maximum impact and maximum efficiency. Here on the video, on the bottom right side, is an example of cavitation formed within the cavitation chamber on one lab scale cavitation generator.
So let me now present a few case studies that we have performed in the latest years using the devices designed into our lab. So we've focused our research on some critical contaminants present in wastewater, meaning various pharmaceuticals and also one personal care product. We investigated several different devices, starting with the Venturi design, in this design, we investigated 6 different pharmaceuticals. We tested the effectiveness of the device in different water matrices, and we also tested the effectiveness with the addition of external oxidants. Then we switched to rotary devices, where again, we tested the device on the same pharmaceuticals as in the Venturi design. And later on, we also tested it for the degradation of polymeric chains of polyvinyl alcohol. Again, we tested different matrices, water matrices, and addition of external oxidant. And at the end, which are some experiments that we are conducting at the moment, we combined our rotary device with gaseous plasma. We also expanded the number of pharmaceuticals. We added some new pharmaceuticals, and we only tested that in a simple water matrix. And every time when we start doing some investigation, we always try to conduct our investigation, some preliminary exams, where we try to determine what is going on. We try to understand what happens. And after that, we optimize our devices to make them more effective and efficient and possible for a scale-up, as also Martin has mentioned. So in this first study where we used Venturi design, we investigated its effectiveness on 6 pharmaceuticals like chlorfibric acid, ibuprofen, naproxen, ketoprofen, carbamazepine, and diclofenac. Many of these compounds you probably all used at one point in your life. And what we have done here is we tested the effects of initial pressure, cavitation time, and Hadva addition. The concentration of pharmaceuticals was 1 microgram per liter and was chosen as these pharmaceuticals are found in the environment in wastewater effluents in these concentrations. We can see here that all the tested parameters have a row, and that the highest initial pressure is the most effective, which can be linked to intensity of cavitation and linked to production of more hydroxyl radicals and thus higher degradation. What you can notice immediately here is that pharmaceuticals are degraded, but are not degraded to the same extent, which once again, we come to the point where their degradation strongly depends on their chemical structure and physicochemical properties. Because the radicals form when the bubble collapses. So this means that the compounds can be degraded where the radicals are located, which means they have to be in the close proximity of the bubble water interface. And if one of the important parameters does not fit to this, which is octanol water partition coefficient, log KOA, it means that pharmaceuticals are not in the place where radical forms, which is then connected to their degradation. And for this reason, some external oxidants like hydrogen peroxide can be added, which then increase the degradation also of these compounds, which are more located in the bulk liquid. What is very important in this study is that high degradation of recalcitrant pharmaceuticals was achieved, which are namely carbamazepine and diclofenac. And this is very important if cavitation is ever going to be used in a real scale application. And to then test how the water matrix influences this degradation, we use two types of synthetic wastewater effluent. What it means here is that besides our pharmaceuticals, in this water also other organic compounds are present, and this synthetic wastewater is similar to the wastewater effluent that comes from real wastewater plants. And we can see here that, we chose the point of the preliminary experiments in wastewater, in deionized water showed in the previous slide, is to determine the optimal conditions, which were like 6 bar, 30 minutes, and the addition of hydrogen peroxide. And in these conditions, we then compared the effectiveness of the Venturi design in these different matrices. And you can see that the matrix has a very clear effect on the degradation, which is then lower because other compounds present in these effluents compete for the radicals, which influences then the degradation of pharmaceuticals. Once again, we can see quite satisfactory degradation of diclofenac and also very high degradation of naproxen. Then we continued our investigation with rotary device. We use the same 6 pharmaceuticals at the same initial concentration of 1 microgram per liter at a 15 minutes cavitation time. You can see immediately that we have lowered the cavitation time compared to the Venturi design. And here we investigated the influence of temperature and of Hadrowitz addition and again of water matrix. We can see that different results are achieved when we change the temperature and Hadrowitz addition. And at a certain temperature, the degradation goes up. But then at 68 degrees, it drops, which is related to the cushioning effect because too many bubbles are present, which then lower the cavitation intensity at this point. So again, in this simple matrix, these parameters were chosen, namely 50 degrees and the 340 milligrams per liter of Hadrowitz peroxide for further experiments, which we once again perform in more complex matrices. But in this time, we took the real wastewater effluent from the Ljubljana wastewater treatment plants. You can see here that the concentration of these pharmaceuticals is ranging from 0.9 to 0.3 micrograms per liter. And we tested here also, we changed treatment time and addition of hydrogen peroxide. And again, we can see that these treatment conditions influence the degradation. And at 15 minutes and 3.4 grams per liter of hydrogen peroxide, we already achieved quite good results, even in more complex water. But by prolonging cavitation time to 30 minutes, the degradation that we achieved was close to the one achieved in simple matrices. Later on, we then switched our investigation to another compound that is present in very high amounts in wastewater effluents, which is a synthetic polymer, polyvinyl alcohol, which is widely used in textile and paper industries and in household as detergent pot. And in addition to conventional microplastic, this water-soluble polyvinyl alcohol poses environmental threat because it is usually washed down the drain unnoticed and unobstructed. And if it is not treated during wastewater treatment, it enters the aquatic ecosystems in estimated quantities of several thousand tons per year. What we have tested here is looking at its molar mass, which we decreased from 124 kilograms per mole down to 1.6 kilograms per mole. And we can see that here we tested it's the degradation of the carbon-carbon polymeric chain, which you can see in the right corner. And we tested it with acoustic and hydrodynamic cavitation. And we can see here that we can always achieve better results with the addition of external oxidant, because when only cavitation is used, the breakage of the chains only goes mechanically. And once we add an external oxidant, the degradation is also caused by hydroxyl radical. So we then have mechanical and chemical effects. And from this tear spectra, you can see these bands that grow in the wavelength around bottom here in around 1750, which means formation of carbonyl bonds in the molecule. And the last investigation that we are performing right now is where we joined plasma and generation of plasma using a rotary cavitation device. First of all, we ignite plasma in the Venturi constriction where supercavitation is formed, and the supercavitation bubble is established using our rotary device. In this novel design, we added some new pharmaceuticals, and these are a few of those pharmaceuticals are included in the watch list and are very critical environmental pollutants also in very low concentrations, namely estradiol, bisphenol A, and 2 antibiotics, trimethoprim and sulfamethoxazole. And once again, on this graph, you can see that all of these pharmaceuticals are susceptible to degradation with plasma. But again, they are degraded to different degree, which is once again, we can see that it is dependent on their physicochemical properties and on their structure. And again, the degradation depends strongly on the experimental conditions. And the best results were achieved at 25 degrees Celsius. What is very important here is, and very encouraging, is that we have achieved a very high degradation of estradiol and bisphenol A, and these experiments were performed at 30 minutes. And this gives us very courage to do some more studies regarding this. So at the end, let's just put together what are our futures idea, what we want to do in the future. Well, we will continue to do some deep investigation of chemical and mechanical effects of cavitation of various compounds, but we will expand the compounds from pharmaceuticals. We will dive into biopolymers and microplastics. We will investigate cavitation characteristics to enhance or attenuate mechanical or chemical effects. All this knowledge we will try to use in the design of novel cavitation devices, and also including and combine them with other advanced oxidation processes such as ozone, plasma, or UV. And we will continue with the development of scale-up devices. So we would both like to thank you for your attention and point out that what is the point of all of this investigation is that something that is put here in this picture will not happen in the future, or that we will try to prevent it. So yeah, thank you very much.
So Sebastian, thank you. We have, Mojca and I have come to an end with the lecture. So if there are any questions, please ask us. Otherwise, from our side, that is it for today. Till the next time.
Thank you very much for the interesting presentation. Yeah, how's it at the side of the students? Any questions? Anything you want to have clarified, explained in more detail?
I guess not.
Thank you for the presentation.
No problem. Thank you.
Everything very, very